Yeah, the history of gene therapy, thinking about it, goes all the way back into at least the early 1980s when we realized we could first clone DNA. And thoughts back then were that this was a powerful tool for us in the future to be able to correct genes that affected humans in terms of metabolic diseases or just genetic diseases in general. So the thinking goes back quite far. In terms of the actual clinical studies for gene therapy, those began in the late 80s and the early 90s. And at this point, the idea was to insert a corrective gene into a person's genome so that their cells would function normally and produce the proteins that were needed for a person to be healthy based on a genetic defect. Along the way, there were some difficulties encountered with doing gene therapy trials. So for a while, these trials were put on hold, and there were many questions about how to do gene therapy, how to assure patient safety, what kind of vectors should be used to transfect cells to insert corrected genes, and that has come a long way in the last 10 years. So today there are a number of gene therapy trials in progress that will help enhance personalized medicine going forward. The major roadblocks for gene therapy in the last 20 years have primarily pertained to a question called insertional mutagenesis. That is, when you insert a new gene into a cell, you want to make sure that it inserts into DNA in a place that doesn't introduce a new mutation to a person's cell, or in a place where it turns on a gene that's not supposed to be turned on in that particular cell, or something where it doesn't insert in a place that affects the levels of the normal genes and the proteins that they produce in the cell. Insertional mutagenesis can be avoided primarily by the design of the vectors that are being used these days. The other big concern is that the previous vectors that were used to transfect cells with the corrected gene, that they might recombine somehow and create other viruses within cells. And it's through a lot of knowledge about the viral structures that are being used to transport these corrected genes into cells that that's becoming an issue that's not of as great a concern as it used to be. CTI is conducting several gene therapy trials at this point. They're very interesting and they're very cutting edge. CTI is positioned well to do gene therapy trials because historically CTI has done very complex studies in populations that are critically ill. Having done those types of studies for the last 14 years, it allows us to deal with the complexity of gene therapy. This is really important, especially when you perhaps are initially taking cells from the person whose genes you want to correct. Their cells, and inserting a corrected gene into their cells essentially becomes a personalized medicine for them. The chain of custody of those cells and that transfection and making sure they get back into the right person is really complex. CTI has experience doing these kinds of complex trials because they've handled transplant trials for many, many years, and there is a lot of complexity and logistics associated with transplantation trials.